I can't get my screen to work, so it's okay, I have other stuff. Can I just get you to go to this part? It's genetic engineering, your textbook. It's the last part. Alright. You have your notes 10 already. Okay. As you slowly prepare yourself, in our previous lesson, we were at artificial selection. Artificial selection. Yeah. The idea that in a generation, there will always be variety. Some may be more favorable than others to us, and we may select for them, give them breed with each other, we will get more offspring with those desired traits. What is desired may not necessarily be favorable for the organism's health. For example, we saw that poor dog. Humans may like a flat face. A flat face to a dog means less space for teeth, less space to breathe properly. Is what is favorable to health always likable to us? Maybe not. Some people just don't like big dogs. Actually, maybe the bigger the better if left in the wild. But if we were to take all those flat-faced dogs, those small chihuahuas and put into the wild, right? God knows what happened. Sorry, no offense to anyone who has a chihuahua. Anyone has raised those dogs? Anyone has a dog as pet? No one? No one? No one? Anyone has cats? Who? What's a terrapin? Ryan has terrapins. Two. What do you call? Oh. What about it? Say your brother's name. Parents, is it the name? The name? What? Okay, so that's artificial selection. But you know what the problem with that is that we need many generations to achieve what we want. In a population, there may be only a few with the trait we want. We have to let them mate to the next generation. And this process is very tedious. We need to go through so many generations to increase the number of individuals with the desired trait in the population. That's the old school method. The new method, scientists have. Hello? Shucks. So it's okay. I feel bad. I feel bad. I did not teach them. Okay. The problem with this is that it requires a lot of time, many generations. Because this individual with the desired allele, we want it to spread in the population, right? So how? New method. Yes, Kayla? What? Me? Or you? Oh, okay. Go. Okay, genetic engineering, the forefront. If we already know what is the desired allele, why not we just extract it out and put it directly into the individuals? That saves all the generations that we need to forcefully breed out. that's the next step that we're going to go into today. How are we able to extract out the gene of interest and clone it into another organism? I say clone it into another organism because we are essentially taking out the OG DNA, we copy it and put it into something else. We are cloning. We're making a copy except putting it into something else. So how do we do that? The focus of today, we're going to look at the tools required to genetically engineer organisms. We're going to look at some examples of genetically engineered organisms. To start today, we're going to look at the tools. We need to somehow be able to extract the DNA. We need to cut it out of the individual, and then we need to put it into another individual. There are a few tools required for this process if you are at the page of genetic engineering. I'm going to list down some of the tools required. Number one, we will definitely need the gene of interest. This gene of interest is present in your desired organism. Let's say you have a plant with a desired trait. That desired trait will have come from this gene of your interest. That's the thing you need to take out first. In other words, the gene of interest is your target. That's the thing you want to take out. How do we take out the gene of interest from the organism? We need scissors to be able to cut. Except the most famous scissors we have in the biological world are enzymes. So we need enzymes to be able to perform the cutting action. We call these enzymes restriction enzymes. I think in your textbook they say something restriction endonucleases. Yeah? Same idea. Your textbook will say, elaborate a little bit more. They say endonucleases. The word endo means on the inside. Nucleases means to cut the nucleic acid. Same thing lah. Okay, restriction enzymes, restriction endonucleases. Okay, so this is our molecular scissors. Oh, what's the role of the enzyme in it? The role of the enzyme is more to cut out the gene of interest. Oh, okay. The restriction enzymes, okay, let me introduce some other vocabulary we use. Okay, I tend to use the word digest. Yeah, okay, but it will make a little bit more sense if we can make use of other vocabulary. Okay. So let me just draw certain diagrams on the other side to represent what I'm saying. Let's say this is the individual's DNA. It goes on and on. But we only really want one part. The gene of interest. That's the one we want. We need to make some cuts at the side. Okay, I'm going to draw dotted lines to represent that this is me cutting the DNA. You understand why I'm going to cut it this way in a while? What I'll end up with is a gene of interest with a little bit of extra DNA at the sides. Okay, this is what you'll see later on. The molecular scissors, the restriction enzymes come in at these points to perform the cuts. Okay, so some vocabulary you can use. The area where the restriction enzyme is cutting is called a restriction site. Okay, so it's nothing big. So the restriction enzymes, you can say cut, you can say digest at the restriction sites. I say again, you could say this in a sentence. You could say, I can make restriction enzymes to cut the gene of interest out at the restriction sites. Okay, so we solve one issue. We can cut it out. This one thing. Next thing, we need to find a way to put it into another organism. We actually have a tool that we use called a cloning vector. Have you seen the word vector before? Yes. Vector is like a host, something to hold something for a while. Just like how mosquitoes are vectors for viruses, right? They contain the virus, then they fly, they take from someone, then they fly to another person, they are the vector, they are the carrier. Heard before the word vector? Yeah. So we need a cloning vector. We need something that is able to hold on to this gene of interest that we have extracted out. We call that cloning vector. What is the nature of this cloning vector? The cloning vector is always some sort of a bacterial plasmid. What is a plasmid? A plasmid is circular DNA. What do I mean by circular? That's not the typical structure of a double helix. The DNA is very special. It links end to end. What do I mean by that? So, normally in other organisms, the DNA goes something like that, right? One strand like that, and then the other strand like that. Double helix. But bacteria actually have DNA organized this way. They have the DNA connected end to end. So that means the nucleotide bases just go like that, and then it links up to the other end. That's why we say it's circular. Therefore, it does not have a 3' or 5' end. It's just closed loop. Maybe I should blend this so you have some sort of comparison. This is a cloning vector. Okay, in a while, I will give us the opportunity to perform this. Here, I've prepared several slips of paper with DNA on it. We're going to try to create a cloning vector with a gene of interest inside. Last step, we need to put this gene of interest into the cloning vector so that you'll end up with something like that. So this is inputting the gene of interest, connecting it to the cloning vector, the bacterial plasmid. There's a name for this particular recombined structure. It is called so the next vocabulary to use, this is called a recombinant plasmid. It means that this is a plasmid that has been recombined with something else. Yes. You're right. Okay. So I have not mentioned that. Right? If you just observe from here to here, we actually need to do one more thing therefore, right? We need to perform one more cut. So actually, you will need a restriction enzyme to perform just one cut. Okay, we're going to try this later on. Of this circular DNA, right? Okay, right. When you make this cut, it will no longer be circular. But once you put this gene of interest plus the whatever you just cut together again, it will be circular again, but just slightly bigger. One last tool. If we cut things, we must stitch it back together. We need a glue of some sort. This glue comes in the form of a, it's not really a glue, it's a tool. It's an enzyme called ligase. Ligase acts as the enzyme that stitches it back together. Because at the moment, it may fit like a puzzle, but we still need to be able to stitch it back together. What bond am I looking at here? Is phosphodiester bond. I need to stitch back the phosphodiester bond that I broke at the very start. And that is carried out by ligase. Okay, so broadly, just need you to learn two new enzymes, restriction enzymes and ligase. Then we introduce some other ones like plasmids and so on and so forth. Correct. They catalyze the formation of phosphodiester bonds. Restriction enzymes break phosphodiester bonds in order to cut. You're going to try page 8 now. I have a real example for you to try. Okay. Yes, I collect cup noodle cups. It's another one of my collections. So you know when I eat cup noodles, right? I don't eat from the cup. Because I want the cup to be clean, right? So I will pull out the noodles and all the stuff, right, and then put inside a bowl. Then I will heat it in the bowl, then I have a clean cup. I have a lot of these cups, all different designs. Oh, donkey, you know donkey, right? Donkey got different cups, right? Then I'll collect. Maybe they got seasonal one, right? I'll go and do that. I did. But I like to collect the ones from Singapore, very easy to find. Okay, ready? In this cup noodle cup, okay, you'll find I prepared a gene of interest. Okay, this gene of interest has not been cut yet, even though it looks very short. Imagine that this organism's DNA is just this long. Okay, the gene of interest is the black part, which you will later on see. Okay, I want everyone to collect one gene of interest in a while. Next, I prepared here a plasmid that is not yet circular. I'll need you later on as you come out and collect one scotch tape, you take it end to end, then you will have a circular plasmid. Okay? And you have the time to circularize it. Okay, so all I need to do is everyone come out now, starting from this side. You collect one of each, then take a little bit of scotch tape so that you can create your circular plasmid. How to create the circular plasmid? You'll find that the straight one at the moment it looks like that. One end has a dark shade, the other end has a white shade. You just overlap the dark over the white such that it's just dark all the way. You understand what I mean? Can I? Okay, so everyone, you can starting from this column, come and collect 2 strips plus 1 scotch tape. Then we'll continue with the lesson. Okay, next column. Last column. Can you create your classmate now? Can take. No. That's the challenge here. Okay, everyone has collected a different gene of interest. Okay? Exactly. Wow, when you're desperate, right, you can do things pretty fast.
That's very true.
So, today is a very busy day. I managed to finish my meetings at 11. No, AM. Then after that, I quickly run to the canteen, grab a bao, then I came back. Then in 15 minutes, right, go and print out, and then I go and sit there and then after I went for lesson at 3:02, then after PM time, then after I come here. Yeah, but then you always like, okay, I want you all to learn in class. Okay. So, here, this is what you should have now. You should have a classmate, you should have a gene of interest. Okay? Okay, this is the interesting part. The reality is that we do not just have one type of restriction enzyme. We actually, as scientists, have a repertoire of restriction enzymes. Yeah? Depending on what the restriction site is available, we will use different restriction enzymes. In other words, restriction enzymes are specific to restriction sites. You can only use a certain type of scissors for a certain type of site. For example, here I present to you 3 different restriction sites. Restriction site X, Y, and Z. There's an enzyme that can cut X, another one that can specifically cut Y, another one can specifically cut Z. The interesting thing about how these enzymes work is that they do not cut just one way down. You observe, they do not cut this way. Okay, instead, they cut in a zigzag fashion. They make one snake out. Okay, these are phosphodiester bonds you're cutting. Then we destroy the hydrogen bonds. Then we cut the phosphodiester bond. So the cut is zigzag in nature. Same here. Phosphodiester bond are snip, hydrogen bonds go away, phosphodiester bond. Same down here. The snips are zigzag in nature because when you do a zigzag cut, you end up generating something called, okay, I'm going to show you now the end result of Z. When we do a zigzag cut, you end up generating something like that. You have overhangs. A, T, T, C, and then it's a G. Notice now I have extra bits hanging out. This is what I call a sticky end. I say it is sticky because this extra 4 that's dangling out, it can hydrogen bond to another sticky end that we may come across. And where might we find other sticky ends? Well, if we make one more snip at the plasmid, then we end up generating two more sticky ends. Understand? Yeah? So, really, the magic of this is that you just need to make 3 cuts. Make 2 cuts at the gene of interest on the sides, and make 1 cut in the middle over here. The challenge for you is to find the correct restriction site on the plasmid to make the correct cut. The beauty of it is that the plasmids that we have come with a lot of restriction sites. Because you never know what restriction site is available for you to make the first 2 cuts. Yeah? In other words, you must use the same restriction enzyme. Only by using the same restriction enzyme will you create complementary sticky ends. If let's say you use the enzyme that can cut X over here, then on your plasmid you use another enzyme that cuts out a different form of a sticky end. There's no complementary base pairing taking place. You try to stick them together. Okay, so the challenge for this afternoon is, I'd like you to look at your plasmid, look at your gene of interest, and I'd like you to try to create a recombinant plasmid. I'd like you to create 3 snips, 3 zigzag snips. Okay, I did not ask you to bring scissors. You can do zigzag tears if it's required. Or you can use a ruler to slowly tear it away. If you have stapler, you could staple them back together to signify ligase joining them together. Or you can grab some scotch tape to make the stick. You can make reference to page 8 of your textbook, but by the end of today's lesson, you should complete and create a recombinant plasmid. I will go around to check. The main thing I want to check is, did you cut correctly? And did you show complementary base pairing when you try to stick them back together? Okay? Okay, you may discuss with your friend right now how you want to make your cuts before you commit to the cut. At the same time, you also want to decide where you want to make your cuts. I mean, I've helped you out a bit by color coding the restriction sites so that you can decide where you want to cut. At the end of it, you should have a nice bracelet, the recombinant plasmid you can wear around your head. Wait. you time to try and error, figure out how to solve this puzzle. I have put 3 restriction sites in your classmate for you to decide where you want to cut on your classmate. If you cut it always down, right, down. Down, right, down. You end up having a overhang on the top. Down, right, down over here. This overhang on top and down. Usually it will yeah. So anything extra, right? Anything of the dream. Just extra. Hello, alright, actually this is nonsense stuff. Okay, double confirm with your friend if you are making a correct cut before you make the slip. Wait, I think you're supposed to cut from top, left, from top down, right, then down. Yeah, zigzag. Right, the enzyme cuts specifically this way. Down, right, down. You cannot, you don't. Yeah, it specifically cuts down, right, down. Yeah? Okay, come on, I'll need a few left. Okay, based on my research, this is how it cuts. You will cut it from, you will do it from here. We need to make a cut here and cut here, and one more cut somewhere in your plastic so that you can insert this. The idea here is that you want to create overhangs like puzzle piece so that you can keep in the end of the day. After you make a cut here. Yeah, that is very average for the SPE cut. But the skill here is that you, after you make your cut, you want to make sure that they can complementary base when they make it. Okay, so you did one cut, so you created one SPE. Yeah, so you have to make a cut here also, right? So at the end of the day, you must be able to. So you need to make a cut down, right, down, so that it can stick. And then this overhang over here will be able to stick. Hey, oh, your classmates. Okay, you'll go ahead and see. If you think you've got your recombinant plasmid, just let me check. Okay, the only thing I'm checking is whether it's complementary based. A to A, the B is T, yeah. So this is Yes, Jing. Ah. Okay, complementary. A to T, A to T. On this side. A to T, A to T. Yeah. Yeah. Correct. So it's really about using the same enzyme to cut the same type of restriction sites. They can create same sticky ends. It's like what line base drawing the phosphodiester bond. Okay, so you just make one cut. Okay, so doing this activity, I hope you appreciate how elegant this process is. One enzyme, 3 cuts. This is what scientists do. In the university, when I performed this process, what I did was to search the data bank to find out what are the restriction sites present on the sites of what I wanted. Then I just go to the lab and find the corresponding enzyme that can perform the cut. I will then find a plasmid that also has the same sites that can perform the same cut. And then I'll just dump them all together in an incubator and let the magic happen. It will cut and they will stitch back together. Yeah. Correct. So it's the enzyme plasmid as well as the gene of interest. Or you can do it sequentially, you should lah. Start by extracting the gene of interest. Say all your gene of interest and put it together with the plasmid and the enzyme. You're right. Actually, then we should really sequentially follow the steps. Actually, the interesting part would be therefore, when we put like this, cut plasmid as well as the gene of interest together, there's a chance that the cut plasmid will just be one without anything inside. So it's trial and error. What happens? Thrown away. Yeah, it's not the gene of interest, it's not our interest. At least when we do it in cells. We don't do it in an organism. Yeah. We take some cells of an organism, then we take out what we want, and then the rest of the cells will die. But we don't harm the entire organism, unless this organism is a single cellular organism. Correct, yeah. The original, now the new recombinant plasmid is longer. Oh, okay. Yeah. Okay, interesting. So as I go around, I think I understand now what Mr. is saying. I think a lot of you have the impression that if I want to slot in the gene of interest, I need to cut out the same segment. Okay, I understand now. Okay, when you look at this diagram, you thought that I extracted out a segment from the plasmid. So what you thought? Yeah, you thought I extracted out a segment from the plasmid, right? But I didn't. I really just made a single cut and then I open it up. Okay? I think once you realize, right, that all you do is a single cut, open up, then you realize how efficient this process is. If I have to make two cuts of, a lot more to cut. Yeah?
Then it might not match.
It will match if I use the same enzyme.
Like if you cut.
That is provided that the plasmid came with two restriction sites. But your plasmid only comes with one. So you cannot possibly create two CPNs. You notice? Your plasmid comes with all three restriction sites. Yeah? So that's the beauty of cloning vectors. Cloning vectors come with many restriction sites so that you can have freedom depending on what is present on the opposite sides of the gene of interest. Anything you like to clarify? Because that's really all there is to understand this last part, the challenge. Yeah? Okay, so good question. How might I assess your understanding of this? Number one, I could ask you to describe in words how you might create a recombinant plasmid. Okay? So you could say, number one, I need to first use restriction enzymes to digest at particular restriction sites to extract the gene of interest. I will then use the same restriction enzyme to cut the plasmid. I will then use ligases to help form phosphodiester bonds to insert the gene of interest. Done. Another thing I may ask is, I may provide you, like what you've done here, DNA sequences, and I ask you, where will you cut so that you can insert in? The reason why I want you to physically cut today is because during an exam, you cannot cut your exam paper to figure out this puzzle. You have to mentally imagine that when you perform a cut, one part will open up. But today you've done that. You physically open up after you cut. Yeah? So remember this experience, because exam time, you don't want to have a few pieces of your exam paper. It's very funny, you cut one part, then you shift one part of the exam paper up. Okay? So how does this relate to genetically engineered organisms? Well, after we have generated this recombinant plasmid, we can take this plasmid and put it into cells of organisms without disrupting their DNA. Okay, you see what we're doing up? So let's say I have a single cell that will eventually grow up to be a full plant. The plant has its own DNA. At the same time, with this plasmid inside, it has an extra package with the gene of interest. The plant cell can use its cellular machinery to replicate its own DNA, at the same time replicate the gene of interest. It can also transcribe and translate its own DNA as well as the gene of interest. So we are really just giving, it's like a software update. Yeah? We do not change the entire software, we just give one more update to it. We give an applet to the cell. So that's what we are doing. That's why we need to create a recombinant vector. The recombinant vector is a very nice package that we can use to upgrade plants or animals. The textbook provides two real examples that's in the market right now. BT corn as well as a salmon I don't remember what it's genetically modified for. Is it to grow better or to reproduction rate? Grow faster also. Okay? We expect you to know the case study so that you can at least give a case study if we ask you for a case study. Would you eat genetically modified corn or salmon? Oh, okay, you're done this in job, but will you? Will you eat? Why not? Okay, why yes?
Because it was modified for a reason.
Okay, and what is that reason?
Sometimes maybe to make it more affordable, sometimes to make it more nutritious,
okay.
Sometimes to increase crop yield or like.
Okay, but then you look at the corn, what is modified for is to be more resistant to insects. But you're going to eat this, Whatever insecticide the corn was producing by itself now, it's not for you, it's for to kill the insects. Why might it have some doubt? Okay, maybe it might cause an unknown allergic reaction. Your body doesn't normally metabolize this extra insecticides produced. Another thing, another train of thought is, actually we have no idea how introducing a new enzyme or a new protein or whatever to the organism may have on the large scale of things. You have seen how within the body, just by tweaking one thing, it can affect a whole lot of things. So we really have no idea. Unless long-term studies have been done to see the impacts of the organism, we don't really know how one new protein or enzyme could have a cascade effect. We really don't know. So that's just one of the concerns that we are worried about. Others, will genetic engineering create division? Well, because it's not a cheap process, could it be therefore those that can afford it can do it? Those that can't will not be able to do it? In fact, people are now doing this. They create genetically engineered plants that may be able to grow better, but at the same time, they made it such that they will not be able to produce seeds. You know what this creates? It creates a structure where the farmers are dependent on the person selling the original plant. Is this fair? What if we start progressing to humans? Oh, that's another story too. There's this movie called it's called Get the Car. Go watch it. This story is a sci-fi story set in a world where every baby is formed through genetic engineering. Every parent will come together and they'll sit down and they'll decide from their own sperm and egg, right, which ones they want to take to create a baby. The storyline of this story is that it follows a character that was born not out of genetic engineering but out of normal methods. But this person has trouble integrating in society. I found this very interesting because the name of this cycle is, it's actually used, they are using the nitrogenous bases. ATCG. That's a. Go watch this movie, it's very interesting. Double T, oops. Ethan Hawke is the star. Okay, that's all. We have come to the end of your biology journey. So you are certified top step on your head biologist. You know everything you need to know for the JH line. When you go to SH, if you are interested in biology, or rather, if you want to take biology in SH, understand that we'll spend a lot of time on the smaller stuff. So for example, if you enjoy inheritance, genetic engineering, you enjoy cell division, all the smaller scale stuff, that's what SH will tend to do. SH veers away from the large things like how the heart works, how your eyeball works. That is reserved for if you go to medicine next time. You study medicine in university, then you will go back to all these big things again. But to appreciate how the big things work, actually you must have a foundation of the small things. That's what you're doing in SH. Okay? So actually I would say if you prefer what you learn in JH4, that is more SH-like. Okay? As far as biomolecules, that's SH-like. Okay, next week we'll start going through your papers. I'll start off with the oldest paper, 2019, MCQs. Then I'll move on to 2020 next week. 19 and 20, just the MCQs. Okay, the MCQs take awfully long to go through. Okay, they have no answer key, just the alphabets. Okay, thank you. That's all. Have a good weekend. Okay, next class.
Thank you and goodbye.
Thank you. Yes.
Wait.